Hello, everyone, and thank you for viewing our poster. I'm Luke, and I will be presenting today. If at any time this audio becomes distracting, please mute, pause, restart, or forget about it at your convenience. And without further ado, I'm going to walk through the sections of the poster, assist with interpretation, and hopefully preempt some common questions. Starting from the top left in our graphical abstract, in the top left of that figure, we have a voltage-gated sodium channel. In response to a stimulus, it undergoes a conformational change, opens the pore domain, and allows sodium ions to flow into the neuron. This creates a charge gradient, depolarizing the cell and causing an action potential. Now, moving right, the endocannabinoid signaling molecule anandamide comes into contact with the cell, and it engages a G protein-dependent signaling pathway. Downstream of this pathway, an effector molecule preferentially binds to inactivated sodium channels, stabilizing their inactive conformation and inhibiting normal response to voltage change. Now, the cell receives the same stimulus as it did before anandamide, but the probability that an action potential and subsequent neurotransmitter release will occur has sharply declined. Voltage-gated sodium channels are of keen clinical interest because of their fundamental role in neuronal signaling, as well as their involvement in numerous pathologies. Drugs blocking sodium channels are utilized extensively in medicine for many purposes, and not just like localized anesthetics, but also treating complex conditions such as epilepsy and depression. Now, many of the drugs that block sodium channels do this via a direct action, meaning that the molecule itself is what associates with and inhibits the channel. And it turns out that cannabinoids have been reported to inhibit sodium channels in a lot of different models, from prokaryotic to human isoforms, so possibly hinting at some broadly conserved mechanisms. So anandamide specifically is an endocannabinoid produced in the brain, and it has been previously reported to block sodium channels through a direct action. However, we found that the effects of anandamide on sodium channels were mediated by G protein signaling, and we performed experiments to better characterize this inhibitory effect. Shifting to our methods, we used whole cell patch clamp electrophysiology to record sodium currents. The schema on the left depicts this in a wonderfully simplified way, and the captured image on the right shows the technique in practice as it looks under a microscope. The idea is to patch into a single neuron with an electrode, manipulate the membrane voltage, and thus elicit sodium channel currents. We cultured these neocortical neurons from wild-type C57 mice of either sex and used intra and extracellular solutions to block confounding potassium currents as well as other confounding factors such as glutamatergic and GABAergic signaling. Now, getting into the data, prominently displayed front and center, we have figure 2. Which, with a variety of metrics, shows that inhibition by anandamide is voltage dependent. That is to say, the rate of anandamide inhibition occurs more quickly at less negative membrane potentials. Part A depicts our voltage protocol, where we held neurons at one of 3 potentials and then stepped them every 5 seconds up to negative 10 millivolts, evoking a sodium current. Those currents are overlaid below, before and after application of anandamide for each of the holding potentials tested. Part B is a diary plot describing how anandamide inhibition of sodium currents occurs over time, with the take-home point being that it occurs much slower at a holding potential of negative 100 than it does at one of negative 60. To further quantify this, we used curve fitting to the individual recordings, which were described well by a square exponential equation. And from this, we acquired time constants of inhibition. This process is illustrated in part C, and those time constants are plotted to a logarithmic axis in part E. Lastly, the fractional block, that is the relative amount of inhibition after 200 seconds of anandamide application, is plotted in D. Okay, so moving on to figure 3. What's the idea here? Well, because these channels become inactivated during long periods of depolarization, a different fraction of channel states will exist at a given membrane potential. So we employed an experiment to ask how anandamide changes the curve describing the relative sodium current in response to a voltage step during these periods of depolarization. The bottom left cartoon shows that neurons were held at negative 80 millivolts before a 100 millisecond pre-pulse at potentials ranging from negative 140 to negative 30. We find in the absence of anandamide that the peak current does not substantively change up until a pre-pulse potential of around negative 80 millivolts. In contrast, anandamide dramatically shifts this inactivation curve, and we see a decline in peak current at potentials as negative as negative 120. In figure 4, we continued to interrogate the effects of voltage on anandamide block by asking whether inhibition could be reversed by periods of prolonged hyperpolarization. Before or after near steady state inhibition by anandamide, done at a potential of negative 80, we used a paired pulse protocol as drawn in the top left, wherein 2 steps to negative 10 occurred between a variable holding period at negative 120. Looking at the first data point on the plot, when the pulses are separated by just 1 millisecond, fast inactivation has occurred, and the measured current of pulse 2 in the control group is only half that of pulse 1. However, within 10 milliseconds, the channels have completely recovered, and the amplitude of pulses 1 and 2 are more or less equal. In contrast, after block by anandamide, recovery occurs logarithmically in proportion to the duration of the negative 120 holding period. This demonstrates that initial block by anandamide can be reversed when the cell becomes more hyperpolarized, and also further demonstrates that anandamide block preferentially favors less negative potentials. Shifting focus to a more mechanistic exploration of the pathway, in figure 5, we were curious as to whether this effect of anandamide was mediated by CB1. Though anandamide has been shown to bind numerous receptors, as an endogenous endocannabinoid, its canonical target would be the cannabinoid receptor, CB1. So accordingly, we used a potent and selective CB1 antagonist, that is AM251, and asked whether the effect persisted when CB1 was blocked. Moving to the top plot, we found that not only did inhibition persist, but the time course of inhibition was more or less indistinguishable, and this led us to generalize our approach by blocking lots of G-protein coupled receptors instead of just CB1. So, without getting too far into G-protein coupling, the idea is that by including GDP beta S in the recording pipette, we were able to inhibit GTP binding to G-proteins, preventing their cycling. The results of this are shown in part C, where the effects of anandamide were completely abrogated when G-protein cycling was inhibited, implying that anandamide inhibition of sodium channels is not via a direct action and is instead mediated by G-proteins. So below are some highlights spelled out from the poster. Thank you for bearing with me through this kind of unusual format, and I hope the event has been as rewarding as possible, given the circumstances. Cheers, be happy, healthy, and thank you for coming to my TED Talk.